There are four groups of eukaryotes that are considered right now. Sometimes this changes, but our book is looking at four. And so they're named Excavata, or the Excavates, the clade, Archaeplastida, and Unikonta. And so these big groups are right underneath the domain Eukarya. They're not really in the kingdom, phylum, class, order, family, genus, species, but they are good groupings, evolutionarily related. You can see there are some dotted lines here, which means that we're not completely sure about those relationships. And you can also see that this is a polytomy, all coming off at the same time. We have some ideas about the actual branching pattern of the supergroups, but this phylogeny is not showing you that. So you will not need to know those kinds of branching patterns. You will need to know the Archaeplastida branching pattern, which we will expand on when we get to plants, and the Unikonta branching pattern here. We will be going with the fact that both the Amoebozoans and the Opisthecons or Opisthoconts are closely related, so you would have those connected to each other. So let's start with the Excavata. You don't need to know all that many in the group. We're mostly focusing on those that we're looking at in the lab practical. So there are three clades. I'm not that concerned about you knowing the clades either. You'll need to know Euglenozoans, for example, Euglena in that, but you would want to know that these are all part of Excavata. Mostly excavates are put together in the supergroup because of their cytoskeleton, and the word excavata or excavate comes from this feeding groove, like to excavate means to dig out, and so that's this feeding groove, but not all of them have that. And then they have these clades inside of there that are all monophyletic. So diplomonads and parabasalids are two of those subclades underneath excavates. They have modified mitochondria. For a while they thought some didn't have any, but some of them have very reduced modified mitochondria. And they live in primarily anaerobic environments. These two are the ones we're mostly concerned about for our class, the Giardia intestinalis, also known as Giardia lamblia, and the Trichomonas vaginalis. So let's look at those two. Here's the Giardia. It's an intestinal parasite. That epithet of the species name gives you that hint. And the things I need you to know about it are there's two phases, two stages of its life. There's the trophozoite stage, which I'm showing here on the left and the right with some different staining. And then there's the cyst stage. You can see the multiple nuclei in all those stages. They almost look like eyes, but those are nuclei. And in the trophozoite stages, you can also clearly see the flagella. In our slides, probably what you'd see was the cyst stage, where you don't see the flagella. This is a very hardy kind of stage where it can survive many different environmental insults, and so that's what allows the Giardia to get in the water and then survive until it gets ingested by some other organism like a human. So giardiasis is what the disease is that Giardia causes. It's an intestinal disease that includes diarrhea, greasy stools, severe cramping, dehydration from the diarrhea, and sometimes nausea and vomiting. And a lot of times people get this from drinking out of streams when they are on hikes, or maybe in whitewater rafting or something along those lines, getting the water into their mouth. You can also get it in public places like pools and things like that, although it might be less common there. And it's passed through feces of animals. And so it's not because the water's polluted or anything, but it's just, let's say, a beaver has the giardiasis in its intestinal tract. It defecates, some of that gets in the stream. It's in the cyst form, so that when the person gets it in their mouth and it gets into their stomach and then their intestine, it'll go back into the trophozoite form and give them an infection. Giardiasis can also be known as beaver fever. I prefer giardiasis, which matches Giardia. And a lot of times when people travel and get chronic diarrhea, especially if they've been doing water sports of some sort, giardiasis is the actual diagnosis of the parasite that they have. So that's the most frequently diagnosed intestinal parasite from travelers and in the US. The other excavate in the parabasalid group now is Trichomonas vaginalis. That should give you a hint where this one resides. So it's in the vaginal canal as well as the urethra in males. That is where the urine comes out after coming from the kidneys to the ureters and then the urethra. Asymptomatic in 70% of people. So just because you have no symptoms doesn't necessarily mean that you don't have an STI, sexually transmitted infection. So this would be one they could screen for. The disease you get, or the infection, is called trichomoniasis from Trichomonas. And we have 3.7 million people infected in the US that are actually diagnosed. You can see again, this is what our slide looks like, that these have flagella too, multiple flagella, and then here's the nucleus on these Trichomonas vaginalis organisms. They also have an undulating membrane, which you don't see that well on here, but that helps them to move through the water, or the liquid, I should say, of the body, in addition to the flagella. The last clade in the Excavata that we'll talk about are the Euglenozoans. Highly diverse. Some are chemoheterotrophs, some are photoautotrophs, some are parasitic chemoautotrophs, so they're varied there. They do have this crystalline rod inside the flagella. I don't really need you to know the two groups here for our purposes, but they are the euglenids and the kinetoplastids. So here's the flagellum. You can see here they've cut a cross section. There's all those microtubules arranged with the two central microtubules, and this is the crystalline rod, which is unique to the Euglenozoan group. The Euglenozoan you need to know for the lab practical and the exam is called Euglena. That's its genus. There are multiple species. This is a stained Euglena slide of fixed Euglena. You can see mostly the nucleus is the primary feature that's visible. In the live Euglena, which we may have looked at in class, you can see it's green, which is a good indicator that it has chloroplasts, which means it likely does photosynthesis. So they do photosynthesis, and actually this red thing here, pink looking, is the eye spot, which is a photosensitive area on the cell, which means that it can detect light. And then to move to the light and to move to hunt, you can sort of see this flagellum here. Looks like a little disturbance in the background, but that's actually the flagellum of the Euglena. And so what it does is it does photosynthesis during the day when there's light, and does hunting. It's a chemoautotroph during the nighttime when there's no light to photosynthesize. It'll dive down in the water and go hunt. And we call that combination of photosynthesizing and hunting mixotrophy. So Euglenas are mixotrophs. Another excavate in the Euglenozoan group are the Trypanosomas. So there are two types, Trypanosoma brucei and Trypanosoma cruzi. Now, there are also other species than this, but these are the two that we're looking at. Brucei causes the sleeping sickness, also known as trypanosomiasis. This word may apply to all trypanosoma-induced diseases, so the Trypanosoma brucei does the sleeping sickness. It's carried, but doesn't affect the tsetse fly. And so that's what we call a vector. A vector is the organism that carries the other organism, in this case the protist, from organism to organism that it infects, so here from human to human. We're looking at this in sub-Saharan Africa, probably underdiagnosed, but we have records of 10,000 new cases a year, and they do have medication, but if you're not treated, it can be fatal. You can see these guys have flagella. I'll show you a slide on the next. On the next slide will be a slide from class, and they have undulating membranes, and obviously these are red blood cells, so they are in the bloodstream. Chagas disease is caused by Trypanosoma cruzi, and this one is carried by the triatomine insects, or kissing bugs. They're blood-sucking. I believe the Trypanosomas pass through their feces, and this one's a little closer to home. 300,000 people infected in the US, 8 million people affected in Mexico, Central America, and South America. other infections elsewhere in the world. And here's an example of what the triatomine insect would look like. This is our slide of Trypanosoma. You can see the nucleus, it's the dark spot, flagellum. This is the undulating membrane. You can see it here as well. And these are red blood cells, so it's in the bloodstream. We can actually watch it swim. So you can see the undulating membrane very well as the flagellum. So these are live Trypanosoma brucei in the bloodstream of a human. Then we move to the SAR clade. SAR stands for Stramenopiles, Alveolates, and Rhizarians. S-A-R. So it's a big group, and we'll go through each of these subclades one at a time. So let's start with the Stramenopiles. The Stramenopiles include the most important photosynthetic organisms in the oceans, and also important for 20 to 25% of the oxygen in the atmosphere. And those are the diatoms. And the ones in the Stramenopiles, we talked about, or will talk about, primary and secondary endosymbiosis. The Stramenopiles arose from a secondary endosymbiosis of the red algae. So here are our diatoms. They're algae, which is a catch-all term for a photosynthesizing organism that is not a bacterium or a plant. So these are Protistan photosynthesizers. Nutrition mode, photoautotroph. Unique structural feature are these frustules, which are sort of the cell walls. They're very, very strong, and they're made of this hydrated silica, which is a very glass-like kind of material. So when we look at them under the microscope, if you see something that looks sort of like glass, that's probably a diatom, and you can see they're very rigid in structure. Some are circular, some are square, some are elongated, some are this kind of boat-like structure here, pinnate. So highly diverse in structure, but they all have this glass-like cell wall. They live in the ocean and some lakes. Phytoplankton are small organisms that do photosynthesis. And like I mentioned, they are 20 to 25% of the global oxygen production comes from them. So their ecological role is primary producer, which is oxygen production. That also means they're the base of the food chain in aquatic ecosystems. And they also can, what we call, pump carbon dioxide to the ocean floor. So because of their shell, their hydrated silica frustule, sometimes they don't get decomposed when they die. And these are, a single cell. So that means they'll fall to the bottom of the ocean, and the carbon will not be released back into the atmosphere because they don't get decomposed. And so this sort of acts like a carbon sink, where it keeps lots of diatoms, it keeps the carbon out of the atmosphere. So there are some ideas about increasing diatom amounts to deal with the rising CO2 levels. Of course, that has its risks because the balance of the ecosystem can be upset easily. Sometimes humans try to fix it, but it makes it worse. So there's talk about that, trying to figure out maybe if you can do that safely. Lots of economic uses of diatoms. So we have diatomaceous earth, which you'll see in class, and it's used in gardening to kill insects. Insects have, how they get oxygen into their body is through these tubes or openings in their body sides. And so this glass-like shells, the frustules, clog those breathing pores, and so that kills the insects. They're also, if you look on the back of your toothpaste, it'll say hydrated silica on it, and that actually means diatoms. So they're using that as a scrubbing agent for the toothpaste. Same thing with the cleansers, especially things like polish, silver polish, a lot of times has diatom shells in it. Pool filters, They're very, very fine. This is also why they can be used in optics for calibrating lasers and things along those lines, and sensor machinery. Because they are glass, when you shoot the laser through it, it diffracts certain ways, and you can actually do measurements and calibrate that way. When we talk about the fossil fuel called oil, that's actually ancient diatoms that did not get decomposed, and then down in the earth compressed and such through the heat and the pressure, turned into hydrocarbons, and we extract them today as oil. So they were actually formerly diatoms. We're also looking to use diatoms as biodiesel, so a renewable resource of hydrocarbons. And that's because they store lipids inside their cells. And so we'd be able to harvest those lipids and use them to in place of fossil fuels, because they'd be hydrocarbons. Some of the algae that's being used in looking at renewable energy, so like the biofuels, a lot of that is the diatoms. One of the problems, though, right now is the industrial scale. So you'd have to have football fields of algae in order to power, a not very large facility, and so they're looking to try to make it more efficient so that you could possibly use this renewable, growable source to get your hydrocarbons. The other group you need to know in the Stramenopiles are the brown algae. Now, there are other things in Stramenopiles as well. We're just looking at these two. So they are all multicellular, and a lot of them are quite large. They are photoautotrophic, nutritional mode. We would call them, quote, seaweeds, but that's not really a scientific term. Just means sort of plant-like, but lives in the water. So land plants obviously live mostly on land. So these guys are protists for our classifications. The fucoxanthin pigment is what makes them look brown. So you need to know that for the lab practical, fucoxanthin. They also have chlorophyll, but this is a specific pigment. And you can see many of them have these floats. In our lab practical, we look at sargassum, which has those floats, which helps them stay near the surface so they can collect sunlight. Sometimes they have holdfasts, which are sort of like roots, but not really. They're kind of analogous instead of homologous to roots. But they can attach it to the bottom of the water. Stipe extends it through the water, and then the blades, which are the photosynthesizing parts, would be near the surface of the water. They all live in marine habitats, meaning salt water, so like the ocean. And you'll need to know they live in cooler water. And the way I remember that is I know the kelp forests are off the coast of California. If you've ever swam in the Pacific, you know it's very cold. And so that's how I remember that the brown algae live in cooler water. So here's an example of a kelp forest. They're very, very large. You can see that they extend towards the sun. Kelp forests provide habitats, so that's an ecological role. Also, because they're called algae, they're our primary producers, so the oxygen production and base of food chains. I can show you.
The Channel Islands.
So you can see how large it is and how many organisms live in those kelp forests. Economically, we can eat brown algae. So laminaria, or kombu as it's called, is in many Asian type soups, like Japanese soups particularly. There's other types of algae, brown algae, that you extract this compound called algin from, which can be used as a food thickener. So if you ever take any kind of fancy cooking classes, you may use algin to thicken up your food there. That comes from brown algae as well. On to the A part of SAR clade, so the alveolates. Classic alveolate characteristic are these alveoli, which is where the name comes from. So here's the cell membrane, and here's these pockets full of gas, and so those are where the name comes from. So I might, on the exam or the lab practical, say, give you a characteristic of an organism that they have membrane-bound sacs under the plasma membrane, and you would know that that means it's an alveolate. Our first group of alveolates we will talk about are the Dinoflagellates. Not surprising from their name, they have flagella, 2 of them. They are unicellular, so this is a single cell, as is this and this. You can see the flagellum pretty clearly here. They are made of cellulose plates. You can see they're quite rigid. And actually you can see here's the plate and there's a groove. That's where the flagellum would be, and there's the other part of the plate. They are also formed, just like the Stramenopiles, the Dinoflagellates were formed from secondary endosymbiosis of red algae, highly diverse in nutritional mode, mixotroph, heterotrophs, and photoautotrophs. The ones that cause the most problems in the environment, we'll talk about in a moment, those are going to be photoautotrophs. They are part of phytoplankton, at least the ones that are photoautotrophic, and they live both in marine and freshwater. So ecologically and economically are rather tied together in this case because the ecological problems that affect human livelihoods, and that's economics. So many are primary producers, the photosynthesizers, photoautotrophs. They can cause what are called red tides, or HABs, harmful algal blooms. And what that means is that there's too much replication of the dinoflagellates. You can see here they're in red, but they come in all different colors depending on the pigments that are in their bodies, because they are photosynthesizers, so they have pigments to absorb the sunlight. This means that there are way too many that have reproduced, and so they, some species release toxins that can harm fish. So there's big fish kills, and they can also irritate human skin. It's not good to breathe them in. They get into seafood, particularly mollusks like bivalves, like clams and oysters, and that can make humans really sick if they eat those. 2 species you need to know that are known to cause red tides, and there are others, but Karenia brevis and Pfisteria. And so both of those release toxins that, again, can harm the fish and humans. On a more positive note, diatoms are essential symbionts of many corals. So this is an economic and ecological. Ecologically, obviously, symbiotic, mutual symbiont relationship. And economically, because coral reefs are a great resource for tourism. Ecologically, they're also habitats. And one of the problems we're having with the warming of the ocean is this phenomenon called coral bleaching. And that's actually when the dinoflagellate is damaged in the coral. Coral is an animal. So it's damaged in there, and then it releases the toxins. The coral animal spits it out, basically, which gets rid of their food source. So the way the symbiosis works is the dinoflagellate makes carbon compounds that the coral animal uses for food. And so once it loses its food source, then the coral slowly dies. And it's called coral bleaching because the color of the coral, in many cases, actually comes from the dinoflagellate, not the coral animal itself. So the coral turns white once those dinoflagellates are expelled. I'll put up this video about coral bleaching on the module so you can watch that. Another economic use or value of dinoflagellates is that some species bioluminesce, which leads to tourism attractions. For example, in Puerto Rico, there's Bioluminescence Bay, which they may or may not let you swim in anymore. And you can see as people disturb the water, the dinoflagellates are irritated, and so they release their own light, basically. So people like to go and swim in those bays, and so that's a tourism attraction as well. Another alveolate group are the apicomplexans. They have this apex that allows them to infect cells like red blood cells. The one that you need to know is the Plasmodium. Vivax is one species that causes malaria. There are others. So Plasmodium goes through this kind of complicated life cycle, part of which, this part out here, is in a mosquito. You need to know specifically that it's the Anopheles genus of mosquito. Kills many, many people. It's one of the biggest problems in a number of countries, including the lowlands of Africa, parts of India, parts of the Mediterranean. And remember, malaria is what's correlated with high levels of sickle cell anemia or sickle cell disease, because sickle cell protects against malaria, and that's because part of the malaria life cycle is in the red blood cells. So the sickled version of the red blood cells doesn't allow these merozoites to infect it very well, and so the person's protected from getting malaria. And so there are a couple stages you need to know. Merozoite's one of them, and I'll show you them here. So this is what your slide would look like. So it infects the red blood cell in a trophozoite stage. Here you can see that it's enlarged, and here's that ring kind of structure here. That's the Plasmodium infecting the red blood cell. Here's another version of the trophozoite. The division happens, and soon the red blood cell is filled with these little infectious cells. It's called a schizont at this point. The schizont bursts open, breaks open, and then it releases the infectious cells, which are called merozoites. So the little dots are called the merozoites. Here's one. This is a trophozoite, and then the schizont is really full of the cells. This is a white blood cell. You can tell because it has this multi-lobe nucleus, and all these are red blood cells around it. Another alveolate group are the ciliates. So these are some examples of different types of ciliates. You can see their common feature, not surprisingly, is that they have cilia. They have it in all different arrangements though. They're all unicellular, and they use these cilia to move around and feed. So the ones that we looked at are the Paramecium caudatum. The cilia here are stained in blue. You can see these macronuclei in the dark pink color, and they have also micronuclei, which the Paramecium are conjugating here, which means they basically form a passage with each other and switch their micronuclei, and that generates genetic diversity and genetic variation without actually sexually reproducing. So they'll just divide themselves in half, but to mix up the DNA, they do this conjugation. They are mostly predators, so here's their oral groove where they eat. The cilia helps push that food into that oral groove in the cell mouth, and then it's digested internally. They're the ones with these contractile vacuoles, which help pump the water out. So when water enters the cell, because the cell is hypertonic to the water, so the cell doesn't lyse or burst, the contractile vacuoles pump the water back out. The R group of the SAR are called the Rhizarians. They are amoebas, which means they have pseudopodia. In this case, though, the Rhizarians, so rhizo means root, so you can think of roots being long extensions, and so that's what their pseudopodia are like. They're thread-like extensions, so all these are the pseudopodia sticking out of this Forams shell or test. There's two groups we'll look at, the Forams and the Cercozoans. There are also, you may hear about Radiolarians in your future classes. They're also a Rhizarian group member. So here are the Forams, or technically foraminiferans. Unicellular, they have a test, which is a shell-like structure made of calcium carbonate. So you do need to know that The diatoms are made of the hydrated silica. Dinoflagellates are made of the cellulose plates, and then the forams have the calcium carbonate tests. They are heterotrophic and sometimes symbionts, so or they live off of symbionts. So there's algae that lives inside of the test that makes carbon compounds that then the cell that is the foram can use that for its food. So it's in a symbiotic relationship with other algae. A lot of them also, when they project those pseudopodia through their shell or through the test, they can catch food and bring it back into their cell. They live in lake and oceans. There are some beaches that are entirely made of foram shells, foram tests. So that would be called marine sand. And those beaches could be a tourist attraction, so that could be an economical use. The symbiotic relationship, mutualistic symbionts, that's ecological. And the limestone formations, ecology, because that's an important geographical feature. It's also economical because it can be a tourism attraction. For example, specifically the White Cliffs of Dover in England, very popular place to go. These really sheer, very, very white cliffs. They're made of foram tests. Here's some other examples of how forams might look. They could look sort of like a snail shell. We saw that they have this sort of multi-lobed look. There's other ones that are much larger. There's a lot of diversity in these guys. And some uses of forams in the fossil record are that they can tell us geological age. And I'll show you these guys on the next slide here. So geologic age, they can be a pollution monitor. So when the forams are unhealthy looking, with these deformed tests, it can tell you that there's pollution in the environment. They are also indicators of mass extinctions. So all these are different species of forams. You can see that in the late Cretaceous period, there were many, many species that exist. But in the early Tertiary, which is a little later in time, there are only a few species that exist. Only 2 in this list have survived through the transition from Cretaceous to Tertiary. So this shows that there's a mass extinction that probably happened here. And in this case, the mass extinction is when the same time that the dinosaurs went extinct. But the forams in the fossil record go from very diverse to not diverse, and that shows us mass extinction probably occurred. We can also look at climate in the past through looking at forams. And so when you look at the isotopes of oxygen in the foram tests, the shells, they can tell you the correlation between temperature and these different oxygen isotopes, which can tell you things about the climate. The other group of Rhizarians we're concerned about are the Cercozoans. And their most important feature is down here with the evolution for our purposes. They have this thing called a chromatophore, which is like a chloroplast in a lot of ways, but we look at this chromatophore structure, it's very different from other chloroplasts, and that tells us actually that it's probably derived from a different primary endosymbiosis event when a eukaryote ingested a bacterium, cyanobacterium specifically. This one got a different cyanobacterium that led to this lineage. So that shows us that the primary endosymbiosis leading to plastids, photosynthetic plastids, probably happened more than once. The next group we'll look at actually are the Archaeplastida group, and they are primarily multicellular. They're all algae, so they're all photoautotrophs. And the pigment in these is the phycoerythrin. An erythrocyte means a red blood cell, you might know that, so phycoerythrin is the red pigment. This allows them to live in deep water because since it's red, you know that's what it reflects, the wavelengths of light, and so that means it absorbs blue and green. Blue light and green light are more energetic. They have a shorter wavelength than red light, so they penetrate deeper into the water, which means that because these absorb blue very well, they can absorb the light that reaches deep water better than, let's say, a green plant or a plant with green pigment in it. They also want to know that they live in tropical waters or warmish waters compared to the brown algae that live in cool waters. They live in various depths, but I mentioned they can live very deep. And the color actually varies with the depth, and it's kind of the different pigments allow them to live in different depths. Economically, this is what your sushi seaweed is, if you will. It's the nori. The wrap part of the sushi is a red algae called porphyra. You can also extract a compound called carrageenan from red algae that helps stabilize food. If you look on lots and lots of food products in your pantry or your refrigerator, you'll probably see carrageenan on that. We also agar, which you plate bacteria on in the lab. That is a polysaccharide derived from red algae as well. Ecologically, being an algae primary producer, and they're also the calcified versions of red algae can be coral reef structures. So in addition to the coral animals, there's also calcified algae there that help make those habitats. In our lab practical, the corallina is an example of one of those that would be a calcified red algae. The most closely related group to plants, land plants, are the green algae. Now you can notice that it's not a monophyletic group, green algae, so that's not a very good name, but they are most closely related to land plants. There's two groups, the chlorophytes and the charophytes. I will break those down further when we get to land plants, but for now, that's what you need to know. The chlorophytes are most of the green algae. They are highly diverse, multicellular, like the ulva that's in our specimens, unicellular, like the Chlamydomonas. We saw Volvox, that's also multicellular, and there are also colonial forms as well. Chlorophyll is their main pigment that appears green. They're photoautotrophic nutritionally. Many of these live in fresh water, which is a little different than the other algae. Some of these are part of pond scum, as are cyanobacteria and euglena. Some even live on land, so one of the very few protists that actually live on land. And then primary producers, some are symbionts, classic examples of what photosynthetic organisms do. And finally, we get to the Unikonta group. So fungi are not protists, nor are animals, but the rest of these groups are. So there's really two clades, the amoebozoan group up here, and then this apisthoconte or apisthocont group down here. One of the types of slime molds is called the Plasmodial slime mold. So this is, you'll find it on dead logs a lot of times. It's on decaying matter. It is not a true mold. Mold is a fungus. These are not fungi, they're protists. And the interesting thing about them is that their body is one huge cell with many, many nuclei in them. So they undergo mitosis to divide the nuclei, but not cytokinesis. And so you just get this massive cytoplasm with many nuclei. They have pseudopodia, that's why they're in the amoebozoan group. They are extending through the dead material, and then they can ingest that by phagocytosis to eat. The other amoebozoan group in the slime molds are the cellular slime molds. So they start as, they live most of their life as single cells until most of the time it's when there's stressful environments, but it can be a number of signals. When they come all together and form these things called slugs, so this is many single cellular organisms all kind of acting as a multicellular unit, then they form these stalks, and then the stalks at the top have spores in them, which are released, and then they emerge, and now you have a new unicellular amoeba. Show you. So you'll see them all coming together into making stalks. So there's all the unicellular, and then they're growing together and making all these stalks, which then will release the spores to the environment, and then they'll be single cells again. So you might hear of Dixostelium or Dicty. It's a classic experimental model that people use in the lab to study this transition from single cells to multicellular organisms, because these guys do sort of both. And then finally, we are looking at the Gymnamoebas. It's also actually looked at Entamoebas. These guys have lobe-like pseudopodia. Amoeba proteus is what we see in the lab. They could also have tubular pseudopodia as well. They use that to capture food. So they eat lots of bacteria, other protists. You might see that happening in the amoebas we have in class. The entamoebas are the parasitic amoebas. So here are some examples in the colon of a human, and that's what's giving them amoebic dysentery. So you might have heard of people getting a parasitic amoeba. That would be an entamoeba that's making them sick. So that's a basic overview. Of the protist groups you need to know, and most of the things you need to know for the lab practical. There's a list online of the specific kinds of topics you should make sure you understand, so take a look at that.